Good morning to all the guests present and online today. I'm Kelly from Biotechnology and Pharmaceutical Industries Promotion Office. I would like to welcome all of you to attend 2024 Master Forum on New Drug Development, sponsored by Development Center for Biotechnology, Taiwan Alliance of Extracellular Vesicles, and Taiwan Society for Extracellular Vesicles, co-organized by Bionet Therapeutics, Bionet Corp, Taiwan Microinvasive Aesthetic Society, and Commercial Times. At the very beginning, let us invite guests from the organizers and co-organizers, as well as the moderator, to take a group photo for us. Let's welcome Chairman of Development Center for Biotechnology, 涂信哲董事长, President of Bionet Corp, 蔡正宪董事长, Vice President of Development Center for Biotechnology, 李才坤副执行长, President of Taiwan Society for Extracellular Vesicles, 周涵怡博士. Thank you so much. Next, let us welcome the speakers and the distinguished guests attending today. CEO of Tokiwa-Bio, Dr. Mahito Nakanishi. Founder of Zymedi, Dr. Sunghoon Kim. Mantra Bio, Dr. Ginny Kang. National Taiwan University, 林中天教授. National Taiwan University Center for Biotechnology, 沈汤荣教授. Please step forward to the stage for a photo. I would like to invite 涂信哲董事长 to stay on the stage.
Good morning, everyone. Our distinguished guest, Tzou-Yien Professor, and Professor Zhou Yi, and Dr. Sunghoon Kim, and Mr. X Mok, maybe on Dr. Ginny Kang, and Professor Lin Zhongtian, and Dr. Li Caikun, and 刚刚我至少拿到微秒里面是吗？谢谢。对。Ladies and gentlemen, good morning. Welcome to 2024 Master Forum on New Drug Development, organized by the Development Center for Biotechnology, DCB, Taiwan. DCB was established in 1984, and DCB has actively promoted the innovation and the development of Taiwan's biotechnology industry, and built a model of academia industry cooperation. Our ultimate aim is to improve the quality of life of the people of Taiwan and the world. To celebrate the 40th anniversary of DCB, we hold a forum today to investigate the future prospect of exosome and new drug development. In recent years, exosome has drawn people's attention as a new technology for drug design. Exosomes are small extracellular vesicles secreted by all cell types and playing intercellular communication doors, which provide exosome playing a key role as a next-generation drug delivery platform. Today, we invite many experts from Japan, Korea, and Taiwan to share their knowledgeable experiences. They will introduce not only the biomedical and cosmetic application of exosomes, but also exosome as a therapeutic approach to diabetic disease. In addition, the preclinical test of exosome drug, ExoTIA, will be also demonstrated. Through this event, we aim to foster the constant interchange of new information and promote the development of exosome technology and related applications, bring new breakthroughs in regenerative medicine. We thank everyone here to attending the forum. We also welcome your suggestions at any times so that we can truly fulfill our strengths, thereby enabling us to promote the vigorous development for the biomedical industry in Taiwan and move forward toward to the global market. Welcome, everyone. Thank you very much. Thank you.
Give an inspiring speech. Taiwan Alliance of Extracellular Vesicles is committed to enhance the international competitive strength of Taiwan's exosomes research. Its convener, who is the host of the forum today, Lin Tzou-Yien 教授, is unable to attend here due to official duties. However, Professor Lin prepared a video to thank everyone. Let's go through the video together.
Dear Dr. Tzou-Yien, President of DCB Taiwan, distinguished experts in exosome from Taiwan and abroad, including Japan, Korea, and United States. My dear colleagues, on behalf of Taiwan Exosome Industry Academia Alliance, it is my great honor to extend my warmest welcome to all of you to attend this symposium. This symposium is to celebrate the 40th anniversary of DCB Taiwan. DCB Taiwan is the first R&D institute in biotechnology, which leads the industry chain as well as provides the key of biomedical development in Taiwan. We have established Taiwan Exosome Industry Academia Alliance last year. This alliance provides a network between academia and industry. In the previous year, we have accomplished several achievements. On June 4th, 2024, the Congress has passed Regeneration Medicine Act. This act has made Taiwan the third nation in Asia that have specialized legislation in regeneration medicine. And this act also is expected to drive the robust development of biomedical industry, including exosome and cell therapy. Finally, I wish this symposium a great success. Thank you.
Thanks to Professor Lin for his encouragement. Next, let us welcome the moderator, Chou-Tien Lin MD Host, for introducing today's speakers. Let's welcome her.
Hello, everyone. Thank you for being here. And as very well said by President Tu, that to invigorate the application of exosome research in the industry, actually it requires the concerted effort from many parts. So in this morning session, we have scheduled 4 consecutive talks, very important talks, covering from fundamental research to manufacturing to end products blueprint. So as the first start, we are very honored to have Dr. Mahito Nakanishi sensei, who is the CEO and CSO of Tokiwa-Bio Inc. And we are so eager to learn from his experience in RNA virus in the iPSC's production. And we hope very much to learn from his experience and to harness this power into the exosome biology. So please join me with a big applause to welcome Professor Nakanishi.
Thank you very much for introduction. Thank you very much for organizers for inviting me to this memorial symposium. This 40 years anniversary in the DCB. So this year is also 10 years anniversary to my companies. So it is very honor to have a presentation at these symposiums. So I want to introduce our technology, stem cell vector. And this is quite useful on cell therapy and also in the gene therapies. I want to focus on the cell therapy areas on this symposium. So until recently, human cell therapy is limited to the cell open transplantation or autologous cell transplantations. So most of the case, cells are not modified genetically or other purpose. However, the finding the iPSC has changed the whole world in the cell therapy world, because iPSCs can be converted to many useful tissues. From also iPS cells can be making peripheral blood cells. We can make an unlimited supply of the tissue cells. More recently, we also directly convert the blood cells to hepatocyte or the tissue cells. And iPS cells also differentiate directly to neurons or hepatocyte or the tissue cells. So the conversion of the cell fate is almost unlimited in this age. And gene delivery and expression system supports all of these cell conversion systems. So back to the a little bit in the history, since the discovery of the DNA is a genetic material, all the gene expression system and gene delivery system is made based on the DNA. So I call this DNA era. However, since the COVID-19, we found the messenger RNA works very well as a vaccine. So this shows that we are now an RNA area. So our technology, cells RNA vector, is one of the vector systems based on the RNA. This system looks like viruses. So it is classified as a recombinant viruses. But the encapsulated molecule is a optimized synthetic RNA germs. So how optimized this RNA is very, very long history, so limited time. But this structure can allow this SRV to efficient delivery of the genetic materials. Also, this optimized RNA germ has allowed a very safe and stable gene expression than human cells. So this is the principle of the stealth RNA vectors. This envelope-like structure has allowed the very efficient gene delivery. Also, more than 97% of the genomic RNA is the synthetic RNA molecules with high GC content. This is the key of these technologies. Because most of the RNA virus has a very low GC content that makes the virus very toxic to human cells. So this optimization allows us space the cellular machinery to detect the pathogens. So in the case, SRV has expanded the limitation of the virus vectors, and at least 10 genes can be installed in a single vector. Also, the size of the total RNA, total cDNA is very huge, up to 15 or 14 kilobases of cDNA can be installed. Also, expression levels can be modulated depending on the purposes. Finally, the SRP gene can be completely erased from the cells by using the various approaches. So all of these characteristics is very, very useful for cell generation, for cell therapy or exosome repertories. So the delivery of the genes is through the membrane fusions. This envelope is bound to sialic acid and fused with the aid of the F protein of the virus envelopes. So only the 5 minutes to 10 minutes is required for gene delivery. So it is quite efficient. So you can see that we can deliver the EZFP fluorescence proteins to cultured cell lines, hematopoietic stem cells, or even very difficult target chondrocyte in the human cells in vitro. And by injection, the rat chondrocyte can be express these genes. So this system works both in vitro and in vivo. The target cells is not limited to humans. Monkeys and rodents and rabbit and chicken is a permissive host animals. In the case of the human cells, there's no limitation in the delivery. Only the one single cell line is resistant, A562. But except these special cell lines, almost all the tissue cells is a target of this system. And very importantly, SRV can get the stable gene expression without chromosomes integrations. Other systems using the DNA as a platform, for example, retrovirus or lentivirus, has integrated the chromosomes 100%. Also, even in the case of the AAV, very little amount, 1% or 0.1% of the genome, AAV genome was integrated to the host genomes. So that is a major concern, the safety issue in the recent gene therapy. However, RNA molecules was not interacted with human genomes, so it is completely integration-free. This genotoxic phenotype is very important safety risks in the future gene therapies. So the genes installed in the stealth RNA emitter is expressed as an independent messenger RNA. So each of the genes, installed genes are making the single messenger RNA and making the different proteins. So it is simply installed many different cDNA. We can get the expression of the cDNA from single vectors. Also, the improvement gene expression system allows us to choose the very different gene expression levels of vectors. CPDP, this is the previous working Sendai virus vectors. The Sendai virus vectors is known to express the genes very strongly, but only in the simple systems to express them very strongly only. However, our latest SRV, version 3, we can choose the various SRV. One of the various SRV was depend on the purposes. For example, making the antibody from CH1 cells, we can choose the very high expression vectors. Also, we are using these middle level gene expression vectors for various kinds of cell reprogramming. The only twofold difference in the gene expression affects the effect of the cell reprogramming. So it is very important to choose the appropriate vectors for purposes. Also, the very weak vectors are put in the gene therapy phase. Also, the erasure of the vector is a very important issue of the SRV. This shows that using the ribo switch in response to chemical compound A, we can get the erase of the vectors within a week to the background levels. So it is a very very useful system to make erase the vectors. We get a patent in worldwide, Japan, US, EU, and also Taiwan. This is a copy of the Taiwan patent in here. So Taiwan is a very important our target in the commercialization. So our company is focusing on various areas. One of the important areas is the cell reprogramming with ectopic expression transcription factors. That increase in the iPSC, but not limited to the iPSC. We are directly reprogramming the hepatic progenitor cells and hepatocyte. Also, we are working the directed differentiation of human iPS cells to neurons. Second most important area is cell regeneration or reverse immortalization. In this case, human cells does not change their fate, but they are regenerated or remarkable immortalization is back to the normal conditions. So in this case, MSC and T cells are targeted with this. Also, some field in the we're working in the gene therapy field with the hereditary metabolic diseases. But I want to focus on these further two topics. So back to the history of the iPSC. Dr. Yamanaka, Dr. Takahashi has identified the iPSC using the retroviruses. So in this case, cells are infected with retrovirus encoding the Yamanaka factors. They are integrated the chromosomes and converted iPSCs. But the problem in using this system for clinical application is retrovirus integrated the chromosomes and they are deactivated after iPSC differentiate. The one was the Yamanaka factor is a famous oncogene, CMYC. So almost all iPSCs differentiated, they are converted to cancer cells. So this are no clinical applications. So people working in integration-free iPSC, so second generation, second sender virus vector and the messenger is commercially available. However, they are suffered in the low efficiency and messenger cannot target the human blood cells. Our technology was classified as third generation in the modality the cell reprogramming. So all of these advantages, the previous approach is recovered. So no genotoxicity, suitable for blood cells, ultra high efficiency, uniform reprogramming, suitable automation, and applicable to iPSC and direct reprogramming. So in the case of iPSCs, this is a principle to how to make the iPSCs. Deliver genes uniformly to tissue cells, and after reprogramming, cells vector genome is completely erased, so we can get the completely integration-free iPSC as obtained. Just to compare to the old sender virus and the SRV technologies, how different this is. We have compared the sender virus with 4 Yamanaka factors and SRV with 6 transcription factors. So you can see SRV iPSC vector, it is quite famous vectors to reprogram the iPSC and making the several number of iPSC colonies in the tissues. But using the same cells and same conditions, we using the SRV iPSC, we can get 100 times more iPSC colonies appear in the tissues. So you can see in the large scale tonight, most 100% of the EGP policies is also expressed in the Taiwan60 good iPS markers. So nearly 100% of the cells are converted these ideal conditions into iPSC. Also, we have developed automatic eraser systems. Automatic eraser system is dependent on the microRNA switch. So after all we need to do is infect the vectors on day 0. You can get to see the gene expression, and after 2 weeks, we can get the iPSC without EZAP expression. And after 1 month, we can get the good quality of the iPSC. So more recently, we are developing more sophisticated iPSC vectors for clinical applications. This vector is expected to be available next year. I compared to the iPSC4. This commercially, we are selling commercially this research grade. We can say that this new vectors, even day 7, we can already get the iPSC colonies. On day 9, those are the many iPS colonies are appeared here. You can compare the efficiency is very different. So we are planning to commercialize these best active iPSC vectors for commercialization in next year. So using these vectors, maybe it is possible to make a clinical grade iPS within 2 weeks. So we are looking for partners to making these machines to make a small scale application of to generate clinical grade iPSC. Also, application with this survey is direct reprogramming to blood cell to other tissues, also human iPSC to direct conversions. All of these approaches, complete erasure of the vectors are keywords. For example, this is a collaboration with Kyushu University, Dr. Suzuki. We are making the we are making a hepatocyte from peripheral blood cells. So these blood cells have quite similar morphology and are expressing the cytochrome 450 for very, very high antioxidants. So second topic is cell regeneration and reverse immortalization of human tissues. One of the very active area on this section is the regeneration of the cancer-related T cells. So T cells are isolated from cancer tissues and regenerated to make the T cells younger and reactivate and then back to the patient's bodies. So this approach is now we are in a collaboration with US companies. Also another approach is of course making iPSC from cancer target T cells. We can make monoclonal T cells. Also this approach is working at Chung-Tien Lin University in Japan with collaboration with us. So this technology is supported by the more efficient delivery to lymphocyte. We are change the envelope structures to meses viruses. Meses viruses has a very high affinity to T cells and B cells. So we can convert these systems to more efficient delivery system to lymphocytes. Also, our interest in the MSC as a target, especially in the adipocyte-derived mesenchymal stem cells. This information you take from the Roche Pharmaceuticals, our collaborators. Morphology of the MSC is such that. However, the limit of the MSC is they cannot proliferate very long. On the passage on 5, they lost the differentiation activities. So we focus on the adipocyte-derived MSC because this is a much advantage compared to the bone marrow-derived MSC, as they have expressed more cytokines and has actively immunosuppressing activity compared to the bone marrow. Also, proliferation is not affected by age, but bone marrow MSC is affected by age. However, the most very important activity, it is proliferation is still limited. So we're installing the SRV in the cell cycle related genes in the single vectors. 3 genes are installed in the single vectors. And you can see this yellow line and the gray line is a vector infected MSC. So they are growing very rapidly without limitation in their lifespans. Very importantly, this phenotype was a MSC infected SIV with 6 cell cycle genes. You can see that most of the morphology is normal. So this is very importantly, the MSC proliferated using this system is not loss of the differential activity. We checked this differentiation to the adipose side. The normal MSC has lost the activity to differentiate the adipose side after passage 11, but in the case of these proliferating MSC, they still hold the differential activity to the adipose side. So summarize today's talk. SRV is ideal tool for cell reprogramming for cell regenerations because SRV is applicable to essentially all the human cells. SRV is the best suitable for gene expression. We can choose the best gene expression levels from the various SRVs. SRV can induce a stable gene expression. This is very important for making the iPSC or cell regeneration, but can be erased after cell reprogramming is complete. And the flexible effective vector erasure systems are available. And we are essentially 4 vector eraser systems. Very much high capacity, number of genes and the size of genes is very almost essentially non-limited. The production system for GMP is currently established. So we are focusing on the clinical iPS SRV for clinical iPS generation. We want to launch this vector in the next year. Also, the SRV for directly making the blood cells to convert to the hepatocyte is now under investigation. Will commercially be for long. Also, SRV for directed differentiations also available upon request. Finally, cell regeneration vectors or MSC is currently investigated. So we are focusing on after after the vector is erased, MSC has converted to normal tissues or not. So we take a suicide sometimes, but it is under investigation. So just I want to introduce our companies. Our company is established in 2014 as a Tokiwa-Bio. This is spin out from the AI of spin out from AIST. And AIST is the National Institute of Advanced Research, Science and Technology. It's like DCB. So we are supported by Japanese government AMEDO. GeneSci project and cell reproduction support was supported by AMEDO. Our company located in about 1 hour from Tokyo city, so you can very good access by highway bus. We are occupied this first floor of these small buildings, and we are actively organized these companies. Thank you very much.
We are so very grateful to Professor Nakanishi because as you have heard, RSV and SRV is like a big brother for extracellular vesicles biology, and in its development and application, we have so much to learn, and we certainly hope to have some common ground to share with each other and to boost each other. So I believe that the audience has many, many questions, but I ask you to hold them until the forum. So Now I would like to introduce our second speaker, Professor Sunghoon Kim, who is also an exemplar of / professional. He is a professor at Yonsei University and also CEO and founder of the Zymedi company.
Oh, okay.
Oh, on stage?
Yeah,
Oh, oh.
Make me more nervous.
But you deserve the spotlight.
Oh, right.
So I will leave the time to Professor Kim.
Thank you for kind introduction. Before I start, I'd like to thank Chris Chai of BioNet and other organizers for inviting me to this exciting meeting, and I hope that I can excite you enough with my stories. So I like to spend some of the time, am I allowed to talk, to introduce my research background, and later part I'm going to tell you how I became interested in exosome science. So title is Novel Bioactive Protein Secreted by Exosome and its biomedical and cosmetic application. So in the year of 2011, the Korean government launched a very ambitious project called the Global Frontier. And that project includes 10 challenging science technology areas, and one of them includes drug discovery. And Korean government felt that academicians should help to boost the strengths of R&D in pharmaceutical industry in Korea, because the Korean pharmaceutical industry is technologically a little relatively far behind compared to other major industry in Korea. And that is very difficult to be innovated by their own activities, and the academicians should help them. So there was a huge competition for the project, and luckily I was selected as a director for this project, and still I'm leading that project. So it was initiated almost now 13 years ago, and supported by Korean Ministry of Science, ICT. And the seed money was $100 million for each project. And one very unique about this project is this is completely independent from academic or institutions. So I can run this project as a complete independent CEO. So, but it's a non-profit research organization. So the whole institution don't belong to any university. In my case, at that time I was a professor at Seoul National University, but it is completely independent so that I can have a full freedom of operation. So with this project, I named the Medicinal Bioconvergence Research Center. And it's been now 13 years, and we have published about more than 300 articles, a similar number of patents, and about 30 licenses. We have established the international and domestic collaboration network so far. We have to start this project from something, and I have compared my project with other drug discovery projects, public, private, throughout the world, how I could make this project unique and distinguished. And I decided to focus on the upstream part. As you can all know, that drug discovery involves several different stages from upstream to downstream. And I realized that academicians are much better in the upstream part. For example, target validation and select the hit and lead, and up until the candidate optimization. This part is mostly done in the academic sector. And downstream part, the preclinical trials, clinical trials, and approval by FDA, this is mostly the industry job. So I decided to focus on this early stage of drug discovery by this project. And then I had gathered annually 25 to 35 PIs who are the expert in various areas and make them work together for this upstream part of the drug discovery. And after 10 years of this Biocon project, that we have accumulated enough interesting pipeline through this platform. And then I have a freedom that I can make a company called Zymedi, which can then relay this accumulated pipeline to the downstream, because although we have $100 million as a research grant, that is still not good enough to bring each pipeline to the drug, because as you know, clinical studies needs huge amount of money in addition to that. And the government money is not sufficient enough to push that to the drug. So I had to make a private sector to make the mutually cooperative situation, so non-profit and for-profit collaboration network to make this system sustainable. Biocon is now completely financially independent. So this is, the Biocon doesn't need any research grant from government anymore because it can run its own royalty. So although this is basic research organization, it's self-sustained. So now they can run together as a collaboration network. So let me go back to the science that I told you. Biocon started this target development project, and I decided not to tackle many interesting target here and there, because by doing so you cannot be the #1 in the world. So we decide to focus on certain area and investigate that into the deep level. And I chose very interesting enzyme family called amino acyl tRNA synthetase, and shortly we call ARS. This enzyme is very crucial enzyme for protein synthesis, and it has been known for several decades as a housekeeping gene. Except for the case of antibiotic, nobody makes this target as a drug because people concerned this is so essential enzyme. If you control this enzyme, you may have a lot of adverse effect. And so I decided to use this one because that is a big challenge, and this is the blue ocean. So luckily, after long standing struggle and trial and failure, now we finally have 3 interesting drugs in the clinical stages. So my company has the antibody drug called Rapabrutog, and that is now clinical phase 1 and supported by US NIH. They recognize this one as a breakthrough drug for pulmonary arterial hypertension and other fibrotic diseases. Beziporocin is a small chemical drug. I provide this information to one of the Korea's leading pharmaceutical company called Daewoong, and this drug is in the clinical phase 2 for IPF, and recently licensed to CSP for China market. FJO-PD-MOD is the synthetase-derived biologic, and this company was founded more than 10 years ago when I worked in San Diego. And so this drug is in the clinical phase 3, and last year it was the Japan market was licensed to Kyorin. So I'm very happy to see finally we have 3 drugs in the clinical phase targeting different tRNA synthetase with a different modality, clearly proving that this tRNA synthetase space is druggable as opposed to the old mind that this enzyme cannot make any drug. So what is ARS? So I'm going to get more to the now basic science and introduce the background of ARS. So amino acid tRNA synthetase in the textbook, everybody know the central dogma, okay? DNA is the blueprint for life, and it's a potential. But the real life is expressed by protein. And someone has to translate the language of genetic information to protein. And this enzyme is responsible for this translation. Okay, so they match genetic code with the amino acids so that gene information can be accurately translated to protein. So that's the textbook theory. But when the Human Genome Project and the Whole Genome Project was actively ongoing, I found very interesting, bizarre pattern of this enzyme evolution. As this enzyme goes from prokaryote all the way to human, they become increased, they pick up more functions to cope with the complexity of the life. So, which means that they are not only doing the essential protein synthesis function, they do much more interesting activities beyond their assigned role for protein synthesis. So it's like, academically, actually, I spent such a hard time to prove this is really the case, because this stereotypic definition of tRNA synthetase as a protein synthesis enzyme is so hardly imprinted throughout the whole life science. I had such a long time to convince them they can do much more activities than assign the role for protein synthesis. And I'm very happy to see now in the academic field that this new paradigm is well accepted. And so the whole field is now changed to this non-canonical role of this enzyme. This is like the evolution of your cell phone. A cell phone is originally has one function, telecommunication, when Bell invented it. Now your cell phone is the platform for your life, right? Without your cell phone, you can't do anything. You cannot pay, you can make a living, you cannot even take a bus, you know. I moved around yesterday in the Taipei, and without cell phone, probably I couldn't do anything. So this enzyme is really becoming the enzymatic platform which can take care of whole system of our body. So synthetase now protect your body from various stress and diseases. Not only doing a protein synthesis, but also controlling the body homeostasis. How do they do that? I found that almost all tRNA synthetases are secreted to body fluid. Of course, your blood has almost all different tRNA synthetases, and you name it, every fluid of your system, body system, has secreted tRNA synthetases. How they are secreted, which one is secreted, really depending on your stress condition, your disease condition. So I thought that the secreted tRNA synthetase would play a crucial role to protect your body from stresses and diseases. And that's the point how you can make an interesting drug. So there are three big questions on the secretion of tRNA synthetase. What is the function? How are they secreted? How you can obtain and purify them? So this is another very interesting basic idea. First, what are the activities of tRNA synthetase? I told you almost all tRNA synthetases are secreted for a specific cellular or stressful context. And some of them, when they are secreted, they make a blood vessel, immune activation, protection from infection, tissue regeneration, stem cell activation, even hair regrowth, immune stimulation, and anti-cancer activities. every single tRNA synthetases are secreted in an idiosyncratic manner and play very unique role to protect our body. A couple of interesting cases. AMP1 is the factor always associated with tRNA synthetases. And I found that the AMP1, when we isolate AMP1 from secretion and treat them to wound, they stimulate the wound closure and stimulate the wound gapping and also increase the collagen deposition. So it is very, very interesting material as a wound healing reagent. And the activity can be retained as short as a 15 amino acid peptide. Now, this enzyme is a very active ingredient for various therapeutics and as well as cosmetic. And I expanded this interest further to see whether this peptide, by the way, can also activate stem cell regeneration. So I checked this one, whether it can also stimulate the hair follicle stem cell. And so I compared this peptide with a minoxidil, which is very famous, the hair growing drug in the clinic. And we compared the activity, and it showed very excellent activity for hair growth. And we have investigated working mechanism. The paper is on the way. So I expect that this peptide will be also useful for hair regrowth direction. Another case, we also found a very interesting peptide embedded in the catalytic region of cystaneal tRNA synthetase. The whole enzyme is also secreted, but the activity is retained in this short peptide region, highly stable, and it works as a strong immune stimulator. This is one of the representative data. This is two different cancer antigen. When we combined our peptide with different cancer antigen, tumor growth is strongly suppressed. So we were encouraged that this peptide will be a very interesting novel platform for cancer and antiviral vaccines in different forms. We can fuse that to FC fragment. We can combine them with the various cancer antiviral vaccines. We can also make a conjugated vaccine. We can put this one in a mRNA platform so that that it can be used as a unique and stable immune stimulatory platform, which can be used for various vaccine area. We characterize this one as for the working mechanism. And what's really surprising about this peptide is very, very high thermostability. When you boil it and refold it, the 100% activity is restored. So when it is combined to various cancer antigen or antiviral antigen, it stabilizes various protein vaccines, which relieves the stresses of a cold chain. So this is another case that we can expand this one for vaccine direction. Next question, how are they secreted? So this is the area how I got involved in exosome. For example, lysyl tRNA synthetase was found to be secreted from cancer cells in the cancer microenvironment. And when it is secreted from cancer cells, it activates macrophage and then bring them to the cancer area so that cancer cells can trigger metastasis. So they turn the immune cells to their friends. And this is the in vivo film we monitor. This is the red fluorescence is the secreted CARS1 in the mouse body. And we monitor this real-time migration of macrophage attracted to this secreted CARS1 region. So this is amazing how strongly CARS1 can attract immune cells to. So this could be a very interesting target against the cancer metastasis. we have now antibody and small chemical against cancer metastasis for that. So we investigated how CARS1 is secreted, and then surprising discovery happened that we found that the CARS1 is secreted through the typical exosome. So cancer cell releases a lot of exosome, and we monitored whether the CARS1 is harbored in the secreted exosome. This is the electron microscopy with the immunogold staining. So the anti-CARS1 antibody is coupled with the gold, and we clearly see the CARS1 is located in the lumen of exosome. And this is CD63, which is known biomarker for typical exosome. So this is located in the membrane. This is located in the lumen. More interesting thing is when we treated the macrophage with the CARS1 harboring exosome, the exosome is ruptured near the membrane of exosome, the macrophage, and releases CARS1. So CARS1 is uptaken to the inside of macrophage. So the exosome is not only carrying out the cytosolic CARS1 out of the membrane, it also delivers that to target cells. So we got this discovery and quite interested. We also check whether it is also the case to the other secretion of other tRNA synthetase. So in this case, the immune cell also releases one of the tRNA synthetase, GARS1, glycyl tRNA synthetase. And when GARS1 is secreted, that activate the K-cadherin, very specific receptor, and that also deactivate RAS-ERK pathway, which is highly activated in the cancer cell, and then leads to the cancer cell death. So this is very cancer specific. So GARS1 can be used as a very cancer specific anti-cancer therapy. We check whether this one is also secreted through the vesicles. And this is the also immunogold staining of GARS1 containing the extracellular vesicle. The reason why I didn't say exosome in this particular case, this particle is much smaller than typical size of exosome for some reason. Another difference is the CARS1 was included in the lumen of typical exosome. This guy is displayed on the surface of this extracellular vesicle. So it is somewhat different from the CARS1 containing exosome. And again, we treated the target cancer cell with GARS1 displayed the extracellular vesicle, and we monitored time lapse of endocytosis of this vesicle. And this one, when it is bound to the surface of the cancer cell, it is endocytosis, and then induce the cell death. So how they are integrated to the target cell is somewhat different from the previous case. And when we treated the tumor containing mouse with the control and GARS1 containing extracellular vesicle, this one show potent anti-cancer activity, not only by doing a direct cancer cell death, but it also attract the immune cells near to the cancer cell, and the macrophage directly also eats cancer cells. So it has a dual activity which can give them a very potent anti-cancer activity. So this story goes on, and this is another secretion of tryptophan tRNA synthetase for the immune activation. In this case, different signaling pathway activate the secretion, and in this particular case, plasma membrane is directly the exocytosed containing the tryptophan tRNA synthetase in the lumen. You can see clearly that the WARS1 is located in the inside of this plasma membrane extracellular vesicle, and this is the biomarker on the membrane. So to make a long story short, we have identified 3 different cases containing different types of tRNA synthetase in different morphology and size of the extracellular vesicle. So when you say exosome or extracellular vesicle, actually they are very, very heterogeneous. So in my case, there is a problem for the clear definition of exosome, how you clarify the size and property and activity of exosome. Depending, I mean, you can use the whole mixture for some activity as well, but when you say the drug development, you have to be very clear what you are dealing with. Otherwise, FDA wouldn't approve it. So I thought that I need more refined technology to separate them in a more homogeneous way. So I get back to the technology and then reviewed how they separate the exosome. And I have some complaints about the current technology because it can easily damage the structure of the extracellular vesicle by pressing or centrifuging or something. So I thought that I need a new technology of separation without giving any physical damage to the extracellular vesicle. And I chose 2 technologies. One is microfluidic technology, the other one is nanomembrane technology. Because in these 2 cases, you don't give any damage to the extracellular vesicle. Both worked very well and they are published. But I chose this latter case because of the scalability of this technology. And the microfluidic technology is also working fine, but if you are working at the industry level, you have to do a scale up. And this is more flexible for that. So we make this structure, and then we gave this technology to the company called Metapore. So they further matured this one to more robust level. Now this technology is available for the clinical or the commercial preparation of the exosome. So this is the final. So Biocon was established, supported by government, trying to innovate technology and drug pipeline to the industry. So we give all the technologies to the current existing industry and also spun off the startups who are specializing for this one. So Zymedi is developing peptide exosome drug. Metapore is developing innovative technology for exosome separation. So we have industry and the public sector working together to innovate the drug discovery, particularly in the exosome field. Thank you for your attention.
Thank you very much, Professor Kim, for your very, very inspiring talk. And I would like to think that as you compare tRNA synthase as the evolution of cell phone, then I would like to think that extracellular vesicle would be like the communication protocol that should be that are advancing simultaneously. Right. So thank you very much. Okay, so now we have a 10 minutes break. So please stick around and come back in 10 minutes. Do we have...
we have the refreshment prepared outside, and
so
the forum will continue on time at 10:45. And please return to the venue on time. Thank you, Dr. Zhou. Ladies and gentlemen, welcome back to the forum. Let us invite the moderator, Zhou博士, again to introduce the speakers. Thank you.
Thank you, and thank you for you staying here with us. Because now we are entering into the application and the industrial viewpoint. And now we are going to have a back-to-back expression by Mantra Bio. We have its CEO, Alex Mock, as well as a very important contributor, Professor Ginny Kang here. And Alex is online, and Kang继之博士 is here with us. So please join me in the big applause to Kang继之博士and Alex.
Thank you for the introductions, and thanks the organizer to give me this opportunity to be here. And for our CEO, Alex, unfortunately he couldn't be here today, so he pre-recorded this video, and we will start with that.
Hello, hi, my name is Alex Mock, and I am CEO of Mantra Bio. Today, myself and Ginny Kang will be giving a presentation about how we've been harnessing engineered EVs, and specifically developing for different indications, in particular a lead program in diabetic macular edema. So first off, at Mantra Bio, we engineer EVs using surface scaffolds to develop multi-specific therapeutics. By implementing a surface chemistry, we can actually create allogeneic EVs that are targeted towards different tissues as well as different drug targets. On the caricature on the right, you can see that one can actually use the EV to target a soluble drug target, as well as target other cell types, such as cell type A or cell type B, and do this in a multimodal fashion. One of the, I think, advantages of an exosome approach versus, let's say, a standard antibody approach is that exosomes have a large compendium of surface proteins that one can engineer. We have effectively thousands of proteins that can be engineered, and this gives an emergent property to this modality that allows you to actually interrogate a wide scale of different disease pathologies that otherwise would be quite challenging. In order to get better at understanding the specific structure and the type of proteins that work well on the exosome surface, we have built a platform called Reveal. Reveal is an AI-driven platform that allows us to engineer this diversity of TEVs. We have a broad IP portfolio established in this platform, and effectively what we do is we use artificial intelligence algorithms in order to structurally predict different proteins that might distribute well and bind well to the exosome scaffolds, and also the target engagement prediction. We've applied this platform to our lead program, which is in diabetic macular edema. This is a ophthalmology indication focused on back of the eye diseases. In particular, in this lead program that I and Ginny will talk about, we are targeting two different pathways, IL-6, which is in inflammation, and VEGF, which is in angiogenesis. And an anti-inflammatory and anti-angiogenic combinatoric therapeutic approach can address a $5 billion market opportunity currently. So before we jump into the lead program, I want to give you a little bit of context on what our platform is capable of doing. The core of our platform is really around a toolbox of EV surface scaffolds. These are chimeric scaffolds that we can utilize to engineer different targeting moieties as well as therapeutic products on top of. These are endogenous, and so we can actually utilize cell line engineering in order to distribute them on the exosomes. So we do not need to use click chemistry or any exogenous methods to get our product onto the exosome. It's actually generated by the cell itself. So this enables us to, again, surface display multiple therapeutics per exosome to engage different drug pathways simultaneously. We can also very finely tune the number of copies per exosome on the surface, which allows us to both use this for a therapeutic fashion or a targeting fashion. And we can toggle the structure, which allows us to do things like complex multimers or agonists. All of this, as I mentioned, is IP protected. And on the bottom, you can see a caricature of how we effectively can append different therapeutic proteins onto the surface of the EV. And on the right-hand side, those are different graphs comparing our scaffold versus competitor scaffolds, such as what's known in the space as LAMP2B, which is a very common competitor scaffold. Now, going into the lead indication, diabetic macular edema is the leading cause of vision loss in the working age population globally. It's a critical unmet need. About 1.3 billion diabetics are forecasted to be on on the planet in 2050. And retinopathy is the #1 complication of these diabetics. So it's about a $5.5 billion market today. It will grow to about $10 billion in 2031. There is a blockbuster opportunity for drugs that can demonstrate superior efficacy versus the standard of care, which is anti-VEGFs like Eylea and Lucentis. The blockbuster opportunity is really focusing on, can we actually target all the key disease pathways? Currently anti-VEGFs are targeting angiogenesis and vascular leakage, as shown in the chart here. Vabysmo, which is a new drug by Roche, is also targeting those same pathways. But what's left on the table and not targeted currently is inflammation and barrier function. And as a result, there's quite a lot of patients that do not respond to the anti-VEGFs, and about 50% respond poorly to the standard of care. So really the unmet need here is a differentiated mechanism of action to enable targeting this incomplete responder population, and as well as an ability to target the back of the eye with tissue tropism. And so that's exactly what we've developed in our lead program in MB113. It's a multi-specific TEV that's targeting IL-6 and VEGF. And at this point, I'm going to pass it over to Ginny, who's going to go through in more detail about our lead program. Thanks for your time.
Okay, so Alex gave us great introductions about the different engineering capabilities and talking about the unmet need of the diabetic macular edema. So now I'm going to share you some preclinical data that we have. All right, so as Alex mentioned, the diabetic macular edema, it composes like very different molecular mechanisms. There are oxidative stress, there is glucose metabolisms, and there are angiogenesis, and also the inflammations. And for us, we are focusing on generating a bispecific EVs. So that is an EV that can carry both anti-IL-6 and anti-VEGF, so we can tackle both the inflammation and angiogenesis signalings. And to do that, we initially, as Alex mentioned, so we have the REVEAL program where we use the AI-driven, so this is in silico simulations to identify what will be the best folded constructs that we could put it on the EVs and shuffle them into the EVs. And then we started with screening. So here we generated the cells in the EVs in the 96 wall plate transfections. And then each EVs will be evaluated by a technology called single vesicle flow cytometry. So here, in these small figures at the left here, where you can see these are all different EVs, and with the y-axis, these are different protein expressions, but it's our target protein expression. So you can see in this case, compared to the negative control, we have these all different EVs. These are the top expression candidates. And when we have the negative ones and then the top expression candidates, we could start to evaluate using the primary potency screening. And this is the data that I'm going to show you today. Specifically, is the STAT3 phosphorylation. So this is a proximal readout from the IL-6 signaling pathway. So we can look at the anti-inflammations. And then for VEGF signaling, we will be focusing on the VEGF-R2 phosphorylation. And again, this is a direct, showing the direct activation of the VEGF part of the signaling pathways. And after that, besides these direct target and signaling activations, we are also building disease-relevant models to show the disease-relevant distal readout. And here I will show you the data specifically on the cell-cell barrier changes, because this is a very critical step in the disease progression of diabetic macular edema. All right. So I just show you that we want eventually to build a bispecific, but we look at it at one at a time. So the first one that we look at is the anti-IL-6 activity. So this is the inhibition assay for the anti-IL-6. And just to orient you, the x-axis is the EV concentrations in terms of the particle per ml, and then the y-axis is the STAT3 phosphorylation. So this black line here, so this is a negative control EV, just have the scaffold. It doesn't have the target protein to target and antagonize the IL-6. So as you can see here, it is flat and showing 100%. It doesn't impact the IL-6 stimulated STAT3 phosphorylation. But in contrast to the control is that we have all different constructs of the EVs that inhibit the IL-6 activity. As you can see, the decrease of the STAT3 phosphorylation in a dose-dependent manner. And among them, the 4 to 5 EV, it is the most potent anti-IL-6 enriched EVs. And next, now we have one side settled down. So now we look into the next side, that is the anti-VEGF activity. So to combine both, before go into the primary potency assays, that we use the single vesicle flow cytomic method to confirm dual expression. Again, this is a relative new technology that we're building in-house. And, but it's a flow data, so the idea you could think about it, it is still a flow cytometric data. It's just instead of flow, is usually single cell analysis. Here we are looking into single vesicle analysis. So for EVs that contains anti-VEGF only, you will only see a single positive populations. And then the other, of course, is a negative populations. But when we engineer to have both anti-IL-6 and anti-VEGF, you actually get a dual positive populations, as you can see here. So again, this is showing us when we engineer our cells to create the EVs, both entities, both important inhibition proteins are put on the single EVs. It's not that you have a positive anti-VEGF populations and a positive anti-IL-6 populations. So this is a dual positive population. And now let's look into the potency assay. So here, previously I'm showing IL-6, and here I'm showing you the anti-VEGF inhibition assay. So again, the x-axis is the EV concentrations in particle per ml, and then the y-axis is the VEGF-R2 phosphorylation. Again, if there is no impact on the signaling, then there will be 100%. So first, to orient you, let's look at the 558 and 490 first. So the purple line and the red line. So it is interesting to note that we, again, back to what Alex mentioned, we have different capabilities of putting one VEGF or one anti-VEGF or 3 anti-VEGF on the EVs. And in this case, we saw that the 3 anti-VEGF, the 558 has better potency than the 490. So the more left shift means you can use less EVs to have the effect, so it's having better potency. But interestingly, what we found is when we combine with the 4 to 5, if you still remember from the last slide, that is the anti-IL-6. So if you combine both of them together for the 4 to 5, 556, the lighter one, sorry, this is a little bit harder to see from the color in this screen, but versus this line, the yellow line, the 4 to 5, 490, then you see a big shift, even though we are putting the same anti-IL-6 to it. So these potency evaluations help us to give a very important feedback back to our protein engineering team to tell them that, see, the protein structures, orientations, the steric hindrance could potentially have a great impact on how exactly engagement when we get into the cells. So we have like a lot of these kind of the engineering, the dry lab to wet lab cycles, And that helps us to generate the next generations of EVs. And further, we have done quite a bit of the CMC optimizations to the downstream process optimizations to get to the final lead candidate selection. So for the final candidate lead, as I mentioned, we started to build disease-relevant models, cell models, to evaluate it. So previously, I'm just showing the early signaling, and then now we're looking into a relevant physiological effects. So for diabetic macular edema, so our disease indications, the breakdown of the blood-retinal barrier and the vascular leakage causing the liquid, the fluid, to leak into this extracellular space and causing this swollen retina. And that's why it's called edema. And then this is also later stage in the disease, and was that space that is causing the patients to have poor visions. And if without treatment, it is going to be blindness at the end. And that's why it is a hard problem in the disease progression. And for us, we are building, the model is called TEER. So TEER stands for transepithelial electrical resistance. So you can think of it in a case that where there is a tight cell barrier. When you give it a circuit with the electrode, it is hard to pass by. So your resistance is high or remain. And that is the black line over here on these models. We are using the ARP19, that is a human retinal pigmented epithelial cell model. So basically the RPE model. So again, over time, it is staying pretty stable over time. But in our stimulation, that is the red line. That is where you can see over time, there are the decrease of resistance. That means the cell-cell barrier breakdown, so the resistance is losing. But very interestingly is when we do the EV treatment from the light magenta here, that is low concentration, to the top that is high concentration, you could see that with increasing EV concentration, it brings the TEER back, the resistance back to normal, showing that it is rescuing the cell-cell barrier. When we look into the data in a different format, again, that is where at the endpoint where you can see there is a clear dose-dependent manner from the beginning to low concentration and then bring it back to the control. So again, the TEER data shows that EV could potentially, like from this data, it is direct evidence showing that EV could help the cell-cell barrier to the normal state, and potentially it could revert the vascular leakage and then preventing the blood-retinal barrier breakdown that is the critical steps in the diabetic macular edema disease progression. And besides showing the physiological data using the resistance, we also validated the data using the molecular analysis. So the left-hand side, this is the gene expression data, and we're focusing on tight junction genes. So the tight junction genes are the genes eventually expressed into proteins that bring the cell together. We look into several, almost 10 different tight junction genes, but here I'm only showing you Claudin 1 and tight junction protein 2, these two genes for the data here. And again, you can see a clear dose-dependent manner of our lead EVs that can bring the changes, the loss of the tight junction expression back to the control status. And besides the gene expression analysis, we also look into the protein staining. And here we look into Tzou-1. So Tzou-1 is a protein name of the tight junction protein 1 gene. And here we are doing the IF, immunofluorescence staining. So with the white color, that will be the Tzou-Yien. Here we can clearly see that with the stimulation of the IL-6 signaling, it become white punctate signals, as you can see from these yellow arrows. And then the signals are much weaker comparing to the control. But with the EV treatment, that you can see that again, you reverted the protein expression, not just expression, but its orientations on the cell, surrounding the cell, that it is forming the tight barrier by this continuous and a strong standing pattern. So this is the molecular analysis data that validated the previous physiological data. So hopefully, these two main data sets, although we have other data sets that I couldn't show you here, but these main data sets convince you the potency of the EVs in vitro. But besides, before moving into in vivo, we work on quite a bit of EV lot characterizations. And we have done quite a bit more, but here I want to mainly show you some key analysis that we did, including look into the particle concentrations. And here, this is using NTA data for representative. NTA stands for nanoparticle tracking analysis. But most importantly, is not just the EV particle concentrations. Sorry, I forgot to mention the left-hand side. This is our lead EV that has our target protein enriched for anti-IL-6 and anti-VEGF versus empty vector controls that we always include to compare. So besides the particle concentration, one other key here is a protein enrichment. Again, our company focusing on engineering the cells to create a target proteins on the EVs. So it is important for us to show that there is a much higher concentrations enriched on our lead EVs versus the empty vector, either in a bulk protein measurements or again, the single vesicle flow analysis measurements. And as you can see here, in this particular lot, we could achieve higher than 80% positive of these target proteins. And then finally, similarly to what I have shown already, that we are using both the IL-6 inhibition assay and VEGF inhibition assays to show that there is activities for our lead TEV versus our empty vector controls. So going into in vivo, the animal data. And here, because we are focusing on diabetic macular edema, and with the current standard of care, as Alex mentioned, is anti-VEGF. So we are mainly comparing our EVs against aflibercept, and that is Eylea. So first, let's look into PK biodistribution studies. The EVs, or aflibercept, they are administered to the eye through intravitreal injections. So it is injected in this yellowish area, and that is vitreous humor. So that is the main space for the administration. And mainly, we want to see how long can EV retain there, and then more importantly is where does the EVs goes in those tissues. So in this left-hand side, for this data to look at the EV concentrations in these tissues, we can see that EV does distribute to retina, that is in green, and then the magenta, that is RPE choroid. So these are the key tissues in the disease for the macular edema, as I showed for the previous screen that you saw. And I think the other important and very encouraging part for us to see is after 10 days, that is 240 hours, 10 days over here, the concentrations of the EVs in those tissues are still above our in vitro IC50 concentrations. So that means after 10 days, for our EVs, after 10 days being administered to the eye, it could still have a concentration that is pretty potent. And the other part that is also very encouraging in this data is in the aqueous humor. So for our EVs in this data, that you can see the EV signal are below the quantitation limit in the aqueous humor. Why it is important? Because aqueous humor, it is the major clearance pathway for the large molecules, like aflibercept. So if you injected aflibercept into the intravitreal administration, it will go to the retinal RPE choroid, but at the same time, it is also cleared through the aqueous humor pathway. So it is encouraging. This data shows that the EVs are well retained in the eye. And as I mentioned, because we want to compare our EVs with aflibercept, and here we use the retina to VH ratio, using the ratio so we could compare both of them because the doses are different and it's hard to normalize them together. And with the black, that is aflibercept, here you can see with our TEV, bispecific TEV, it has a higher retina to VH ratios over time. And with this very encouraging PK and biodistribution data, we move into the efficacy analysis. So here to look into efficacy, as I mentioned, the angiogenesis, like the blood vessel growth, that is a critical part for diabetic macular edema. And here, normally for people to look into angiogenesis, the key animal model is called the laser CNV, the laser-induced choroidal neovascularization model. The idea is using the laser to poke holes at the back of the rodent, in this case is usually rat eye, and where you can see the lesions start to grow out. The bigger the lesions means the more the angiogenesis. And for us, that we want to see the EVs or your drugs can reduce the lesion size, that means anti-angiogenesis. So for our EVs, the lead EV, this bar, where you can see it does reduce and it is statistically significant lower than our vehicle control. So this is showing that EVs does have efficacy in anti-angiogenesis in this model. And how good it is, here, let's look into the aflibercept. So this is, the red line is aflibercept 200 microgram per eye. So our lead TV is as efficacious as this 200 microgram Eylea dose. And what is this dose? So this dose in the rat, because the rat eye is much smaller than human eye, so we need to do those scale down and comparison. But this number is actually 5 times to the now standard of care, the 8 milligram Eylea dose. So this is a very encouraging data to us. And again, showing our engineering capability, we always put the control here. And in this study, we see much higher efficacy in our lead EVs as comparing to the empty vector control. So at the end, so this is 2 years of development in Mantra, and we have went through the cycles of engineering, production, QC, preclinical in vitro, and preclinical in vivo. We have done like more than 1,000 constructs, made more than 5,000 different products, do a lot of different QCs, including purity, particle size, protein, as I show you, different kind of protein analysis, charge analysis, and also aggregates analysis, especially for us because we're putting a very high concentration dose to make sure there is no aggregations in our formulation is pretty critical to show for our final product stability. In vitro side, I show you that our EVs can inhibit both the anti-inflammations and anti-angiogenesis pathways, and we could restore the barrier disruptions. What I didn't get to show you is we also using in vitro studies to identify tox species because we want to make sure that there is a clear target engagement. On the in vivo side, I show you the PK biodistribution data. Although previously we also work on other mechanistic data, and then I also show you that the EVs get superior efficacies and superior retinal localizations in our in vivo data. And but there are still a lot that we need to do before the final IND filing. But for all these great efforts and data, I would like to finally acknowledge my team, especially the leadership team, Alex, Maria, Jen, and Ollie, and also very much appreciate all the hard work from my team, that is the preclinical biology team, Lisa, Florence, Julian, and Wei Chen. And I'm going to stop here. Thank you for your attention.
Thank you very much, Dr. Kang, and also Dr. Mok, for sharing their experience and showcasing the importance of a tailored engineered EV coupled with the power of single EV screening. Okay, then now, please welcome with me our last speaker, Dr. Lin Chung-Tien. Lin Chung-Tien is a professor at the Department of Veterinary Medicine at the National Taiwan University. And he's going to talk to us about the dry eye condition in animals and how he proposes to tackle with it using exosomes. So, Professor Lin.
Okay, thank you. Okay, thank you. President Tu and Dr. Chai and all guests and all audience. Today, I'm honored to attend the forum to exchange knowledge with so many experts in this field. Today, I'd like to share some finding of the clinical finding of exoteal and dry eye-caused corneal lesion in a rat model. Dry eye syndrome, also known as keratoconjunctivitis sicca, is characterized by decreased tear production and with the result increased tear evaporation, leading to ocular surface discomfort and damage. Dry eye can result in damage to the ocular surface, increase the risk of ocular infection and corneal abrasion or ulcer. It can impact vision and quality of life. It's a persistent and frustrating condition that usually require long-term and continuous measurement. There's an urgent demand for the development of innovative therapies. This illustration showed us the tear film composition. The tear film covers the cornea and protects the cornea. It is composed 3 layers. The outer lipid layer and the middle aqueous portion, and the innermost is the mucin layer. Each layer provides an important function. The lipid was produced by the meibomian gland of the eyelid, and the aqueous is produced by lacrimal gland, and mucins produced by the conjunctiva corpus cells. There are a number of functions of tear film. Briefly, provide oxygen and nutrients, and antimicrobial infection, lubrication, removal of debris, and provide immune hemostasis. So it's key for the health of the cornea and conjunctiva. So deficiency of the tear film, regardless of etiologies or type, it causes dry eye syndrome. It is multiple pathology changes of cornea and conjunctiva. Dry eye is the chronic progressive disease causes ocular discomfort and vision impairment. The cost of dry eye patient measurement is huge. In the U.S. healthcare system, it costs US $50 to $60 billion per year. And the incidence of dry eye syndrome in humans range from 5.5% to 40% around the world. So it's a big health issue in humans and also in companion animals. In veterinary medicine, dry eye is one of the most common ocular disease in the dog, leading to severe keratitis, pigmentation, and secondary infection, corneal abrasion, and ulcer, and also cloudy cornea, inflamed cornea, and the neovascularization. So it causes a lot of corneal change and corneal damage. In young dog, they can have a dry eye because of a genetic defect. In old dog, also have the dry eye because the degenerative lacrymal function. And also in brachycephalic dogs, these dog breeds with big exposed eye and short nose, like a Pekingese, Pug, Shih Tzu, British Bulldog, French Bulldog. They are prone to dry eye and corneal inflammations. And there are two major types of dry eye in companion animal: qualitative and quantitative. The quantitative dry eye is deficiency of the aqueous portion, and the qualitative dry eye is deficiency of lipid and mucin. The aqueous portion may be normal. Dog with qualitative dry eye is more frequent with the quantitative, about 70% versus 30%. And both dry eye type cause corneal inflammation and damage. And different dry eye etiology in companion animal cause the same outcome, the corneal inflammation and the damage. For qualitative dry eye, usually we evaluate with the ocular surface analyzer. There are numerous research regarding the pathophysiology of the disorder to allow us to know better about the complex disease. Dry eye syndrome has complex etiology and pathophysiology. This diagram is a simplified version. Intrinsic factor like aging and hormone imbalance and autoimmune disorders, dysbiosis, and systemic anticholinergic disparity may lead to tear film abnormality and corneal inflammation. Hence, the beginning or extension of the corneal surface, desiccation, barrier disruption, granular secretion dysfunction, neurosensitization, and apoptosis of corneal cells. The homeostatic dysfunction of cornea together with the tear composition disruption cause the unstable tear film. At the same time, the extrinsic factors such as low humidity environment, wearing contact lens, or post-surgery, such as ID surgery, post-ID surgery exposure, and the LASIK surgery. It causes increased tear osmolarity and activates stress signaling pathway. The inner inflammatory mediator CD4 positive T cells are activated, and this a lot of change to make the forward mark the dysfunction of the immune regulation of the cornea, and the dry eye affected cornea become inflamed and develop corneal damage and injury. There are a number of functions of tear film to the cornea. Thus, the deficiency of the tear film develop multiple pathology of cornea and conjunctiva, especially the inflammation and the activated immune response in the cornea and the conjunctiva. Inflamed cornea are prone to damage and cell death. and corneal abrasion and ulcers occur commonly. Current treatment for dry eye-associated keratopathy, including the anti-inflammatory and immunomodulator, tear replacement, and antibiotic. The anti-inflammatory, including the corticosteroid and non-steroid anti-inflammatory drugs, tetracycline, derivatives, immunomodulator, including cyclosporine, tacrolimus, and corticosteroids. And artificial tears are important to keep good moisture of the dry cornea, but it requires frequent administration. Antibiotics usually we use for the ocular infection and the corneal abrasion. So these are used in clinically. However, these are limitations of the current treatment for the dry eye. There's no cure for dry eye despite of increasing availability of pharmacotherapeutics. And dry causes multiple pathology of cornea and conjunctiva, but most medicine provides a single major function or indication. And in dry patient, lifelong and frequent medical management is often needed. In severe dry cases, limited efficacy or poor response to medication is a common outcome. And the side effect or risk of long-term medication you have to consider. For example, in corticosteroid, the risk of raised intraocular pressure, glaucoma, or secondary bacterial infection, or corneal melting, and antibody resistance, cataract formation, delayed wound healing, and in non-steroid anti-inflammatory drug, corneal staining, burning, delayed wound healing, etc. And in immunomodulator, cyclosporine, tacrolimus, low penetration into the ocular tissues cause eye irritation and vision blurring and slow onset. Mesenchymal stem cells has come into view in recent advance in dry eye therapies. The goal of the mesenchymal cell in dry treatment is to regenerate new tissue to repair the cornea, conjunctiva, and the damage caused by the dry eye. Dry eye causes corneal inflammation, corneal abrasion, nerve damage, excessive immune reaction, affecting patients' vision and quality of life. And stem cells, mesenchymal stem cells have anti-inflammatory, regenerative, neuroprotective properties. Scientists have explored the expression in corneal repair. Okay, so we've been trying using this stem cell to treat dry eye in dogs, and found this beneficial in treating the dry eye. Now we move to exosome. Okay. The exosome are vesicles produced by mesenchymal stem cells. They are nanoparticle size, so it's high permeability and relatively stable compared to living cells, and have low tumorigenicity compared to cell therapy. And the mesenchymal stem cell-derived exosome are used as therapeutic entity as effects of immunomodulation, anti-inflammatory, anti-apoptosis tissue engineering, and the nanovesicles is an ideal drug carrier as well as biomarker. Though there are still some challenges, such as purification of exosomes require high technical demand, the study on the toxicology and treatment efficiency has been ongoing worldwide. So the exosome has the advantage of high permeability, stability, and low tumorigenicity, which compensate for the limitation of the mesenchymal stem cell treatment. And exosomes can freely pass through the various biological barriers without blocking microvascular circulation, and making them an ideal drug carrier. And the exosome is considered non-immunogenic, so it can be used in clinical application on a large scale. Okay, in order to investigate the efficacy of mesenchymal stem cell-derived exosome on dry disease, we conduct preclinical tests of exoteal on treating dry eye caused corneal damage in a rat model, and evaluate the efficacy with several clinical, ophthalmic, and laboratory ways. We perform surgical excision of both extraorbital and infraorbital lacunar gland in SD rat. This mimics the condition of severe dry eye syndrome. It can reach the maximal reduction of tear production from normal 11 to 30 millimeter tear production to the 2 to 3 very low tear production. And the tear production reduction is constant and permanent. And also in this model, more prominent expression of clinical manifestation of dry eye syndrome, and it's reproducible and predictable dry eye syndrome and the corneal change. In this preclinical study, we label the surgical day of lacunar gland removal as day -2. The baseline data of clinical ophthalmic and the imaging analysis was obtained, including tear volume measurement and slit-lamp biomicroscope, and fluorescein stain, corneal, and optical coherence tomography to evaluate the corneal morphology and to measure the thickness. Then the surgical excision of the infraorbital and the extraorbital lacunar gland was performed. Full range of clinical and the image evaluation was performed again 40 hours after the surgery. And the day was labeled as day 0, which is confirmed established of dry eye in this model. At the same time, the beginning of exoteal treatment. The clinical ophthalmic and image examination were performed every 7 days until day 56. And the tear volume assessment was performed throughout the study to validate the dry eye status of this animal model. And also in this study, there was a subgroup of in which rats were euthanized at the day 7 and day 14, 28, and 56. To harvest ocular tissues for histopathology, immunohistochemistry, apoptosis assay, and the expression of cytokine gene. So this is just a group and timeline for ocular tissue harvest for the laboratory analysis. Rats are divided in 2 major groups. In the control group is induced dry eye without any treatment, and the treatment group is the exoteal, topical exoteal, BID twice a day to the KCSI. The preclinical test of exoteal and dry associated clinical lesion were evaluated in 4 major ways. including clinical ophthalmic and imaging, including the clinical ophthalmic imaging assessment, histopathology, immunohistochemistry, apoptosis assay, and the expression of cytokine. Okay, in clinical ophthalmic assessment, we use the slit lamp to evaluate corneal health and lesion, and we look at the corneal neovascularization and measure the blood vessel length, and we use the fluorescein stain for corneal ulcer abrasion and measure the stain area, and use OCT to analyze the corneal morphology and measure the corneal thickness. In histopathology and the immunohistochemistry, we evaluate the structure of the lesion of the cornea at a histology level, and analyze the location and severity of corneal inflammation, especially leukocyte infiltration count. A label and count myofibroblasts and fibroblasts to look at the corneal remodeling and the scarring, and to do the apoptosis assay for the corneal tissues by tunnel, and to analyze the cytokine expression by qPCR. This is the corneal angiogenesis. We look at the reaction to the corneal damage and inflammation. So this is dry eye without treatment. This is treatment with Exotear. So the green line is the treatment group, and black line is the non-treatment group. So both group has increasing corneal angiogenesis since day 0. The Exotear treatment group shows reduction of angiogenesis after day 14. A small rebound was noted in day 35, but the blood vessel continued to regress and keep in low level. And the control group had persistent develop more blood vessels. Until day 21, before it tapered down. However, from day 42, the control group developed a big rebound to develop a lot of new blood vessels all the way to the end of study. They make a big difference of angiogenesis in the, between the control and the Exotear treatment group. And we do the fluorescein staining of the cornea to look at the corneal abrasion or ulcer. The fluorescein staining of the cornea is to identify the unhealthy corneal surface and indicate the corneal abrasion ulcer. The highest level of fluorescein staining of the cornea in the Exotear treatment was on day 0, down 0. Then since the treatment of Exotear, the fluorescein staining area is substantially decreased throughout the study, and the control group cornea stained prominently between day 7 and 21. The fluorescein stained area are larger in the control group than the Exotear treatment group for most of time in the study period. It indicates the Exotear has great protective function to the KCS affected cornea. The KCS-caused corneal abrasion was much faster repaired or restored with the Exotear treatment. And we use the OCT to evaluate the corneal morphology and to measure the corneal thickness. So we can know the reaction to the corneal damage. So after the induction of the dry eye, the control group had epithelial thickening for lasts for 28 to 35 days. So it's very big thickening and reaction. While Exotear treatment group also think is significant by day 7, but the the thickness declined subsequently and remained minimal thickening after day 28 until the study ended. And the thickening trend of the corneal stroma was noted similar in both groups. The white blood cells were mainly infiltrate in the corneal stroma. Both groups have similar trend of decreasing white blood cell infiltration from day 7. However, the exotier treatment group showed almost 50% less leukocyte infiltrate than the control group throughout the study. It indicates the corneal inflammation in exotier treatment group is much less than in the control group. The myofibroblast and the fibroblast count is to look at the corneal remodeling and the scarring. And the exotier treatment group had a steady and gradual increment from day 7 until day 28. They ensure a linear reduction until the end of study. As for the control group, there was a late start of corneal remodeling after day 14, then much increased myofibroblast infiltration in day 14. And from day 28 to 2056, you can find this 1/4 higher myofibroblast count than the exotier treatment, leading to a much more corneal scarring in the control group. And the apoptosis of the corneal tissue, you can find there's also much higher level cell count of apoptosis cell in the control group, in the untreatment group, than in the exotier treatment group. And we collaborate with BioNet team to conduct molecular analysis inflammation marker in the KCS cornea with or without treatment with the exotier. In the left figure, you can see the expression of pro-inflammatory cytokine, IL-1 beta in the control group 10-fold higher than the exotier treatment group. In the right figure, the expression of pro-inflammatory cytokine tumor necrosis factor alpha in the control group was 2.5-fold higher than the exotier treatment group. From the data, the expression of pro-inflammatory cytokine was much more suppressed in the exotier treatment group than in the control group. And this is also the expression of the cytokine. So from this figure also, the anti-inflammatory cytokine was more activated in the exotier treatment group than in the control group. So that's the summary. Taking together the finding of the preclinical test of exotier and KCS-led corneal lesion in the rat model, I summarize into this table. In the control group, KCS caused corneal inflammation and the neovascularization was last for 28 to 56 days, and all the way to the end of study. And the exotier treatment group, the anti-inflammatory property and so immunomodulation was significant in the exotier treatment cases eyes. And the case caused the corneal inflammation was suppressed, and the minimal level of corneal inflammation was noted in the exotier treatment group. And secondly, in the control group, we found severe and prolonged corneal epithelial abrasion or reaction to the damage during the study. And in the exotier treatment group, was found to have excellent protective function to the KCS affected cornea, and the dry eye caused corneal abrasion was much faster repair or restore following treatment with exotier and minimal reaction to the damage. And thirdly, about the corneal remodeling and the scarring, in the control group, the late corneal remodeling process and persistent and severe corneal injury trigger a high number of fibroblasts and myofibroblasts, which contribute to increased corneal scarring. In the exoteric treatment group, stable and early corneal remodeling process from day 7, and gradual and limited increment of the fibroblasts and the myofibroblast number to minimize corneal haze scarring in the exoteric treatment group. And fourthly is apoptosis of cornea was considered high throughout the study, and the exoteric treatment cases cornea was protective from the programmed cell death triggered by cases caused corneal inflammation. Okay, the summary is that the findings are encouraging. And based on the result of ophthalmic and laboratory evaluation for the preclinical test of exoteric in the dry model, the topical exoteric with multiple function has demonstrated great potential for the treatment of dry caused multiple corneal lesion and inflammation. Okay, thank you very much.
Today we are very honored to have many experts from Japan, Korea, and Taiwan to share their experience and insights in talent development of Exosome. And now the floor is open for discussion. And I would like to introduce the host of panel discussion today, Vice President of Development Center for Biotechnology, Li Caikun 副执行长, and also invites CEO of Tokiwa-Bio, Dr. Mahito Nakanishi, founder of Tzou-Yien Dr. Sunghoon Kim, Mantra Bio, Dr. Ginny Kang, and National Taiwan University Lin Chung-Tien 教授. National Taiwan University Center for Biotechnology, Shen Tanglong 教授. Please step forward to the stage. Thank you.
So I would like to first thank the staff and also the speaker for giving wonderful things today. And now floor is open for questions. I guess I will let you guys to ask. First, we do have some question to ask our speaker here. Anybody here? Yeah.
For this wonderful talk, I'm Jialin Xie from the DCB, and I want to have the specific question for Dr. Kang, yeah, and Zinni, yeah, and because when we talking about use the targeting exosome, especially we have the like a surface scaffold for the molecule expression. So in the future, if you want to use in the clinical or the IND, do you need to characterize how many molecules on the exosome surface? And also in this the conjugation, I don't know what's the engineering methodology you have. Is the conjugation is the homogeneous in the each exosome? I am talking about the single exosome. Do we need or do we just use the like the bulk expression for this the issue? Thank you.
Thanks for the great question. So, couple of aspects. So I think I want to clarify a couple of things. The first thing is for us, it's not conjugation. So we engineer the cells to produce specific proteins on the EV surface. As Alex mentioned at the beginning, because we care a lot of the original protein, like those tetraspannings, integrins, they still have their important cell engagement functions. It's just we add extra proteins on it. If there is exogenous conjugations, you might interfere with all those protein-protein interactions. And for the characterization part, yeah, I totally agree with you. It would be super important to look at it in the single vesicle level, and then that's why I show you quite a bit of data on it. We developed the co-developed the single vesicle flow cytometric analysis, and that really allows us to really quantify how many target proteins on each EVs. And as I show that, I think we get to like 400 or 500 copies per EV. It is pretty high comparing to other competitors. And we get to very high efficiencies. Like our QC criteria, release criteria is actually, it needs to be higher than 80% to be positive for those EVs. But again, like I think it's still very important to remember, like what we are deliver the drug at the end, it will be a bulk dose. So the bulk analysis is equally important, and that's why we always report it both. Thank you.
If here, I think we have a couple of also the EV expert here. If you have anything to add to related questions, you are welcome to do so as well. So I have related to the questions actually on, for example, you are using the bispecific. So have you demonstrated that the bispecific actually is greater than just two mixture of the IL-6 and maybe EGFR and so on and so forth? Because it's a great engineering technique, but it might be a tool or in the application or in clinic consideration and manufacturing point of view. Maybe can you elaborate the advantage of that?
Yeah, I think that is also a very great question, and that's something that we kind of like debate in our company over time. But I think at the end, again, going back to giving a drug dose, if I guess right now, I don't know if there is really a limitations. Maybe with future CMC formulation advancement, you could squeeze how many EVs that you like, then potentially you could have like half of them to be anti-angiogenesis and half of it to be anti-inflammations. But at least for our company, we do face the issues that we can't really squeeze enough EVs. There is also only a certain limitations on that into one single dose. Imagine for the eye administration, we are talking about 50 to 70 microliter, so it's a very small dose for human. And in the rat, that is only 5 microliter. So how many EVs can you squeeze in the 5 microliter is what we're doing day in and day out. So in that case, having both moieties on single EVs could be a great advantage. But again, that is for us, it may not necessarily is needed for other disease indications.
Any of you have any comment? Tang Long?
So my name is Tang Long Shen, from National Taiwan University. But actually, I have a question for Professor Kim. Okay, so you are talking about those R's, and there's a different R's. And do you think they have shared some common functionality in anti-tumor, also the immune suppression or some immune modulations? And that's the first question. And the second question, so you see or you found those R's can be secreted by the exosome and play some functional role. So, but the mechanism you are mentioning actually is after secretion and target to the cancer cell, then will be squeezed out and be endocytosis. And why it's to me it's a little bit weird. It's why not just the tumor cell uptake the whole exosome containing those R's?
Yeah. So first question, that are there any commonalities among this tRNA synthetases when they are secreted? And I think so, and they work together for one function, but the working mechanism appeared to be different. So which tRNA synthetases would be secreted really depends on cell context. So their working mechanism is different, but ultimately they work together to control or protect the bodies. Second question, why not taking whole exosome or vesicle inside? And that I don't know. We found two different cases for endocytosis, as I mentioned. GARS1 displayed extracellular vesicle case. The cell take up whole vesicle. And GARS1 is working as a targeting molecule from that case. In the case of GARS1 containing exosome, the exosome appeared to be ruptured. I don't know why. But when it approaches to the target cell, it releases GARS1 right at the front of the cell membrane, and the naked protein is uptaken. So I think, as I mentioned in my talk, all these vesicles appear to work in a different way. And that's why we have to be a little more careful to understand the working mechanism of these extracellular vesicles.
So you also implicate probably those different arts have a different regulatory mechanism to be screened.
Yeah.
Via exosome, is right?
Right. But although all those different pathways and mechanisms for secretion mechanism work together for one function. See what I'm saying? Okay. And coming back to your bispecific vesicle, same thing, that I tried to use this GARS1 to engineer the extracellular vesicle to target, because its target is cancer specific. We try to use this one as a targeting molecule. When we squeeze in GARS1 on the EVs, that depending on how much molecule you display, the physical property of this vesicle appear to differ. So you have to be careful how many molecules you want to display on the surface. And that part probably you need some optimization before you launch your prep to the clinic. Because depending on the numbers of molecule, physical property of EV may change.
Yeah, in this part I also want to, I'm sorry. So I will also want to comment. So when we always express some protein or molecule in the cell and try to make the cell secrete exosome containing more engineering protein or molecule, and because it's overexpressed, so we don't know what happened is really the intact protein really come to really lock into the exosome or not. And when we overexpress, so to cell is probably a kind of overloading. So that's why always when we overexpress the protein in the cell, you can see those overexpressed protein can get into the exosome. That's because it's too much protein that's unwanted. So I will try to remind everyone that exosome originally, until now, is still served as the garbage trash, garbage bin. So when you try to culture those cell, particular stem cell, and use a very rich medium, and the exosome you isolate, many of exosome actually still is like a garbage. So be careful to do this way, and don't believe, okay, all the exosome you isolate from cell is all functional or active. Sometimes it will damage. There has some problem, in the health.
Can I comment
Sure.
on that? I want to say totally, and I also want to emphasize, I think in our company, that is why we believe that scaffold is important, and then also simulation is important, because we have tried numerous times that, again, it's not just you overexpress and that it will just naturally fold it correctly and then display it correctly on the UV surface. It's not that simple. So that is something that we put in a lot of efforts and weight on it. And also talking about yeah, like we also see the same thing because you are changing the machinery. You could potentially generate, like let the cells secrete a lot of EVs, but not necessarily all the EVs carries the ones that is potent. So having the right purifications to get the right prep is also very important in our process.
Okay, so I think should come to the questions since we have, I think you've been in United States, we have Korea, we have Japan, and also Taiwan expert here. So I was thinking about related question is how's the regulation right now for example, for the EV right now or for the engineering EV and the nature EV. What's the regulation now in Japan? Could you comment on this? And then we follow with the Korean and so on.
Yeah, I'm not so familiar in regulation EV in Japan, but probably it is expected to be quite soon. It seems the same regulation is decided, but it is not currently. It is not decided. Yeah.
Well, to my knowledge, that the Korean government set up the first guideline of therapeutic exosome for CMC preclinical and clinical trials 5 years ago, and that was quite restrictive. So they revised it and announced the revised version the last year December, which is more progressive. And so, but unfortunately, so far there is, to my knowledge, there is no exosome therapeutics in clinical studies in Korea yet. And in the cosmetic area, that probably the culture soup containing exosome, as long as they show they completely remove the cell and cell debris, that can be used as an ingredient for cosmetics. So, but for the clinical area, there is not a single case yet in the clinical study, and but industry expect that it will be open soon, because some exosome therapeutic companies doing a clinical study outside of the country.
So I think for the FDA's perspective, EVs are is new, but it's still a drug, so it still falls into the. So FDA has like two big categories. Like one is like the small molecule drug or like regular drug, and the other is biologics. And I guess if you consider like EV itself having functions on it, and it is considered as a biologics, so it follows the biologics regulatory parts. But I think I also heard about a discussion that, so especially for people if they're interested in using EVs just as a delivery vehicle and then they have other drugs inside, and EV itself does not have a function, then you could consider the other path. But I think it is probably going to be very hard to prove that EV does not have a function because it is like naturally, I guess, going back to what Professor Sun said, like whether it's a garbage or it is a real treasure, like it still has a function in certain way. So I think I see like FDA like looking at it as a biologics and then follow those parts still, like building your assays, building your CMCs to the final product.
So I think Taiwan, because it just passed that, so I don't think we need to discuss in here. So because the time is quite limited, so I will have the final questions for the panelists here. Now, from your aspect, what's the strengths in Japan? For example, in regenerative medicine, I think your technology is wonderful and so on, and how that can collaborate with Taiwan in the angle of yours.
of course, it's very interesting to collaborate in the Taiwan companies and Taiwan academia. Maybe our technology is quite useful for many aspects of the regenerative medicines. So currently, many Japanese scientists has given us an idea that provide the idea to how many, for example, the hepatic proliferative cells, hepatocyte generation is their. They offer us their idea. They have a transcription factor. They identify the transcription factors, and we can provide vectors with their transcription factors. So any kind of the genes or interest can be installed in this system currently. So many proposals is very welcome. Also, we can have a proposal to Taiwan companies and Taiwan academia. So I welcome to have a connection to Taiwan companies. Thank you.
I believe that the exosome science has a great potential for novel the modality for the new therapeutics area. And since it has a lot of promises, at the same time, it has also a lot of challenges to prove, to get past the FDA. And I think no single lab, no single scientist, no single country can deal with these challenges. I think we have to work together. And Taiwan has great companies and communities who are open for the collaboration. And exosome, you need content also, so the exosome expertise should work with what to deliver, how to purify. Combining all those three questions, then I think exosome platform will emerge as a new modality in the future. That I'm pretty sure that will happen soon. For that, we have to do some tight international collaboration.
Yeah, I think I totally agree with all what have already been said. It is a lot of development needs to happen, and like for example, in our company, like we need to build our own characterization method. As simple as that, because there is just not really great analytical out there for that. So I do see like collaborated with different space and being very open-minded and consider all different people with different background to bring in their own expertise will really help to move this field forward.
Professor Lin, you have anything to add? Yes, okay. Kang Long? Quick.
Well, I try my best. Yeah, it's a new modality of the EVO called the exosome, and in this field, and particularly regeneration medicine, I believe there's many things we still need to be explored, including the scientific aspect, like biogenesis and the functionality, the cargo loading, and deliver specificity, and so on. And definitely have to integrate other effort, like in regulation and also the mass production industry, and how to really make the link to the indications. And so, but all of this, I guess, in the East Asia, including Taiwan, South Korea, even Japan, we have the niche in the, we have the very good CDMO niche, the experience from those electronic industry. So after isolation, you need an automation machine, and after isolation, you have to categorize by a lot of the equipment, and those equipment is not so cheap, right? It's expensive. So I believe many laboratory actually have a lot of debt because buying those machines.
Okay.
But we can do those though. So
we have Kang Long too.
Yeah. So it's a good niche for us to have the experience from those electronic industry. So to build our so-called CDMO and to because we have a lot of talent, so we can combine this effort together and to really help the whole field in research and also industrialization to go on?
With that, I would think first, DCB is under the Ministry of Economy, and we will try to integrate things. And also, there's two society. One is for the Industry Alliance in Taiwan for the EV, and also the society. We have the president here, Dr. Chou, and also the former president, Tang Long, in the academic sides for the society. And those are very important. And Taiwan, it's been beautiful that although we are small, but we are very tight, integrate together. And so with that, I will thank everybody for the participation and also the panelists for sharing your expertise and so on. Okay. So I will close the section. Thank you.
Many thanks to Fu Zhi-Xin and all the distinguished guests. That's really wonderful. And I would like to express my appreciation for your attending today. And the event has come to the end. Please remember to take your belongings with you when you leave. Thank you. Wish you have a wonderful day. Thank you.